Hi, welcome to this edition of Class in the Kitchen, Fermentation in Your Favorite Foods. I'm Katie Lee.
And I'm Doja Cooper.
And I'm Abby Anderson.
Who likes bread, pickles, and chocolate? I do.
Me too.
Me too. We've got a treat. How these favorites are made. You may not know that microbes are responsible for some essential roles in making these foods and their magical taste.
We hope you're as excited as us. The first topic is our ever-popular sweet treat, chocolate, and the involvement of microbes in its development. As you can see, we have chocolate laid out here for my Hershey Kisses and hot cocoa. It usually takes 5 to 7 days to ferment cocoa beans. Without the process of fermentation, chocolate flavor would not exist in the actual cocoa bean. During fermentation, there's a reaction of compounds that enable the chocolate flavor to be prevalent. It all starts with the harvesting of the cocoa beans, then breaking the pods and preparing them for fermentation. Fermentation occurs in 2 steps and in 2 places. The first step in fermentation is the formation of sugars into alcohol and then to lactic acid, commonly known as vinegar. In more detail, a microbial succession occurs with yeast, lactic acid bacteria, and acetobacteria, and activates the fermentation of pulp sugars. As you can see, we have a diagram of called the anaerobic phase, because the pulp does not let air circulate. Yeast isn't just for bread. The fermentation of yeast transforms the pulp sugars into ethanol.
Now tell me that isn't eco-friendly.
Of course it is. The production of ethanol is an exothermic reaction that generates energy needed to produce lactic acid. The pulp then breaks down and drains away, leading to air penetration. On the 3rd day, the increase of air causes the growth of the aerobic bacteria, acetobacter.
Sounds like a transformer to me.
Yeah, well, it does. The acetobacter transforms the alcohol into acetic acid. This is an extremely exothermic reaction that can increase the temp up to 50 degrees Celsius. Wow, that's hot. The acetic acid penetrates into the bean, and with the high temps, the internal compounds begin to mix and react with each other. The reactions result in the beginnings of the chocolate flavor. Now we're getting somewhere. To get that smooth, chocolatey taste, here's what happens. First, the proteins are degraded by enzymes to polypeptides and amino acids. Reducing sugars like glucose and fructose are released. Part of polyphenesis is oxidized and combined into large tanner molecules. Part of the polyphenols and caffeine are diffused and leave the cell, lowering the natural bitter taste of the cocoa beans. Chocolate shouldn't be bitter when it makes us so happy. On the 5 to 7 days, the fermentation process comes to a close and the drying process begins. The temperature is lowered. The amount of moisture must be reduced from around 47% to 7%. Now that we've got our beans fermented and dried, the next step is roasting and enjoying. Nature never intended for us to have the joy of eating chocolate because it's the result of the cocoa bean dying. So enjoy every little morsel.
One of the basic items any chef must know how to make is bread. It has been a solid part of human diets for thousands of years. As a baker, I know that I appreciate the actions of yeast in my bread making process because it makes the dough easy to handle, increases the amount of gas in the dough so the bread doesn't turn out flat, improves the final texture, provides a nice taste, and extends the life of the bread. All important to any baker. Yeast uses fermentation. It starts by converting the complex carbs in the flour into simple sugars, then it begins to feed on. Almost instantly, it releases carbon dioxide and alcohol, which are important byproducts. You can speed up the process by increasing the temperature between 75 and 85 degrees Fahrenheit, or you can slow it down by cooling. Remember that yeast will be in high temps above 140 degrees. When the carbon dioxide is released, it gets trapped in little air cells in the bread scone and is elastic gluten strands. Gluten is created when using the wheat flour and water. The two combine and two proteins, gliadin and glutenin, mix together to form gluten. When you are mixing the dough, the gluten forms strong crossing and parallel bands that are very similar to rubber bands. The more you knead the dough, the more complex your protein web is. Besides holding the carbon dioxide release immediately, the gluten must be strong because it has to hold the leaving gases that the yeast releases over a period of time. As the carbon dioxide and leaving gases are released and the gluten expands, that's where the height and lightness of your loaf of bread comes from. When your bread is baking, the alcohol burns off and tenderizes the gluten strands, leaving the delicious flavors and aroma. So just a quick summary for all my bakers out there. When protein, starch, and fat molecules break down into their small building blocks, amino acids, simple sugars, and free fatty acids through fermentation, we are left with wonderful, tasty, and smelling bread.
Hi, every time I see a jar of pickles, I can't help but think of the Little Rascals song, I got a pickle, I got a pickle, I got a pickle, hey, 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 well, whatever. It is so interesting that we can turn a regular cucumber from our garden into a completely different treat, a pickle. This is a process that can happen with fermentation. To make pickles, you need to take the cucumbers and place them in a container. In the old days, it used to be a barrel, and it would take 4 to 6 weeks for it all to happen. In this container would be a solution of brine, which is made up of salt and water. Lactic acid bacteria, here we have it, is commonly known to grow on the surface of vegetables. The high concentration of salt inhibits other microbes from growing. At this point, the fermentation of the skin of the vegetables turns it from green to yellowish green, and the insides are usually white into translucent colors. The lactic acid bacteria digests sugars in the cucumber and produces lactic acid. This lactic acid is responsible for the tangy taste of pickles, and it also slows the growth of spoilage bacteria. Since the lactic acid bacteria is munching on all the sugars, it starves bad bacteria that may try to grow and ruin your treat. The amount of salt you put in your brine is very important. If you put too little, bad microbes might grow and ruin everything. If you put too much lactic acid bacteria, it may allow more salt-tolerant yeast to grow more quickly, ruining the batch. Interestingly enough, salt is responsible for the nice crunch when you bite into a pickle.
Hope you enjoyed our show. Tune in next week for another episode of Class in the Kitchen. Bye.
Have a banana, Anna. Try the salami, Tommy. Give it to gravy, Davy. Everybody